What if I told you that every function in your body, movement, sensation, even your thought, start from a simple microscopic unit? In this video, we are going to break down the cell, its structure, its organelles, membrane dynamics, transport mechanism, and even how cells communicate, divide, and die. And by the end of this class, you will understand this in a way that is clear, exam ready, and impossible to forget. In medicine, confusion costs marks and mistake costs lives. Stop memorizing, start understanding. This is Dr. EBM, where medicine finally makes sense. Alright, so first off, what is a cell? Now, a cell is the structural and functional unit of all living things. In a simple term, a cell is simply the basic living unit of life. Now, you should understand the hierarchy of life. So, usually in the hierarchy of life, we have atoms coming together to form molecules. Molecules form macromolecules. Macromolecules form organelles. Organelles are in the chemical level. So it is these organelles that form cell at the cellular level. Cells form tissues, tissues form organs, organs form system, and when all the system in the body is working, it forms a functioning living organism. So the cell sits at the center of all of these levels. So it's very important you understand cell physiology. Cell physiology. So what is a cell made up of? The composition of a cell. Cell is made up of, number one, water. About 70 to 85% of the cell is made up of water. Then you have ions like potassium, calcium, phosphorus, magnesium, sodium. Then you have lipids, mainly phospholipids and cholesterol. Then you have proteins that play a structural and functional role. And you have carbohydrates. Now, it is all these components together that come together and allow the cell to maintain a structure, generate energy, and perform specialized functions. Alright, so moving on, we'll quickly talk about cellular organelles. Now, as you can see on the screen, there are a lot of cellular organelles, a lot of them. So you have the cell membrane or the plasma membrane, you have the lysosome, the smooth and rough endoplasmic reticulum, peroxisomes, cytoplasm, Golgi apparatus, the nucleus, the ribosome, and the list goes on and on. So first off, we'll start by knowing or emphasizing on that all cell organelles have a membrane except for ribosome, cytoskeleton, and centrioles. This is an exam question. Please note that all cellular organelles have membrane, a limiting membrane, except for ribosome, cytoskeleton, and centrioles. So quickly, let's talk about the cellular organelles, their structure and function. So first thing is the nucleus. So this is the nucleus, right? The nucleus is made up of the nucleolus, the nucleoplasm, and the fluid inside is the nucleosoil. So the nucleus contains chromosome that contains the chromatin material, and inside it you have the DNA. Now, the nucleus is surrounded by a nuclear membrane. Now, all membrane have the same structure. We'll talk about the cell membrane later on. And inside this nucleus is the nucleolus. The nucleolus is important because it is the site of RNA production. So it's a site where there's ribosome formation and it's very active during cell division. It's a dark staining zone under microscopy. Very important, very important. Alright, so next on we'll talk about the endoplasmic reticulum. Now the endoplasmic reticulum is two. There's the rough endoplasmic reticulum, which you have here. You can see on the surfaces are rough compared to the smooth, which you can see here. The rough is rough because ribosomes are attached to the surface. So the rough is also called granular, while the smooth is agranular. So the rough is for protein synthesis, while the smooth is for lipid and cholesterol synthesis. Emphasis. Next on is the Golgi apparatus. Right? The Golgi apparatus is responsible for carbohydrate synthesis, for modification, and packaging. So we say it's for packaging and repackaging of protein molecules. Very important, into the vesicles. Next up, quickly we'll talk about the mitochondria. Now, the mitochondria is generally referred to as the powerhouse of the cell. The powerhouse of the cell. So here you have the mitochondria, you have the mitochondria. So the mitochondria has a double layered membrane. Remember, there are two organelles that have a double layered membrane. The nucleus and the mitochondria. So the mitochondria has two membranes. The inner membrane is regarded as a folding known as the cristae. So the general function of the mitochondria is to produce ATP, to produce energy for the cell. Hence we say it is the site of oxidative phosphorylation. Also take note that there are two places in a cell where you have DNA, the nucleus and the mitochondria. So the mitochondria and the nucleus have a lot in common. But the DNA in the nucleus is called chromosomal DNA, and that's why the nucleus is called the brain of the cell, because it controls all cellular activities via the DNA. Check out our videos on DNA and RNA transcription and translation. But the mitochondria is hardly controlled by the nucleus because it has its own chromosome. Rather, it has its own non-chromosomal DNA. Hence, the mitochondria is regarded as a mini cell. Next up, quickly, we'll talk about the lysosome. The lysosome. Now, the lysosome is referred to as the garbage system of the cell. The garbage system of the cell is also referred to as the suicide bag. This is because it contains hydrolytic enzymes or digestive enzymes. These digestive enzymes are responsible for intracellular digestion. Hence, it is responsible for destroying all worn out cellular structures. Moving on, we'll talk about the centrioles. The centrioles are responsible for cellular divisions. Remember that a pair of centriole is called a centrosome. Remember again that a centriole lacks a limiting membrane. You have the cytoskeleton. The cytoskeleton is regarded as the skeleton of the cell and it maintains the cell shape. Also take note that the cytoskeleton lacks a membrane and the cytoskeleton is made up of microtubules that maintains the cell shape. Now, we'll talk about the cell membrane. The cell membrane. Now, the cell is surrounded by the cell membrane, and the structure of the cell membrane is described by the fluid mosaic model proposed by Singer and Nicholson in 1972. Now, what did they say in this fluid mosaic model? The fluid mosaic model describes the structure of the cell membrane as a lipid bilayer, a phospholipid bilayer with an hydrophilic head and an hydrophobic tail. The fluid mosaic model that we are discussing describes the cell membrane again as a phospholipid bilayer. So you are seeing two layers. This is layer 1. So you are seeing two layers. This is layer 1, and this is layer 2. So the hydrophilic head is pointing outwards, while the two hydrophobic tails are pointing inwards. And this is because the body is made up of water. So the head that loves the water is pointing out towards the water, while the tail that hates the water is pointing in, away from the water. So fluid mosaic model describes the cell membrane as a phospholipid bilayer 1 and 2 with hydrophilic head and hydrophobic tail. Now this arrangement allows for selective permeability. The lipid is permeable to fat-soluble substances. So it easily allows fat-soluble substances, organic substances to easily pass through. Now there are three components of the cell membrane. You have lipids, proteins, and carbohydrates. Lipids, proteins, and carbohydrates. The lipid is predominantly phospholipid and allows fat-soluble substances to pass because it is the largest component. Then you have proteins. These are the proteins. There are two types. You have integral protein. This one here is also called transmembrane. That's because it transverses the entire cell membrane. You would understand this when we get to transport. And then you have peripheral protein. Peripheral protein here is only found on the surfaces. Peripheral, not deep, on the surface. Integral, inside. So what are the functions? They act as channels, they act as carriers, as receptors, and as enzymes. Then we have carbohydrates. Now the carbohydrates that are found on the cell membrane are few in proportion and the least as compared to lipids and proteins. Now these carbohydrates exist in conjugated form. So they exist as glycoproteins and glycolipids. Usually they are regarded, the lining, the surface is called glycocalyx. The glycocalyx provides negative charge and helps in cell-to-cell recognition. So the functions of the cell membrane are selective permeability, cell communication, formation of resting membrane potential, receptors for hormones and drugs, cell identity in blood group determination and antigens, and then it maintains cell size and shape. You can go over it to hear them once again. These are the functions of the cell membrane. Moving on quickly, we'll talk about cell junctions. Now, there are three types of cell junction. There's tight junction. Now, this tight junction, from the word tight, do not allow substances to easily pass through. Tight junction. They are found in the kidney and the intestine. You have gap junction. Gap junction allows substances to pass. They are found in the heart. Then you have desmosomes. Desmosomes are found in the skin. They prevent tearing apart. Then we'll talk about cell communication. Now, cells communicate in various ways. Usually autocrine. Autocrine communication describes when a cell acts on itself. Paracrine communication is when information from a cell acts on another cell. While endocrine, paracrine is via diffusion, autocrine, auto on itself. Endocrine is via hormones. So when information is sent to different cells via hormones, that's endocrine. Now when a cell has done its job, it is physiologically removed from the body, and it can also be pathologically removed. So cell death occurs via apoptosis, and necrosis. Apoptosis, one, is programmed cell death. Programmed cell death. It is a controlled process that involves caspases. The mechanisms involve DNA fragmentation, chromatin condensation, and membrane destruction. Necrosis, on the other hand, is unprogrammed cell death. So necrosis is usually pathological as a result of a disease state, unlike apoptosis that is physiological. Necrosis is also called cell murder. Now note that when cells multiply abnormally, that's malignancy. So cells have a regulation, they are supposed to divide and stop. But if they continue dividing, it causes malignancy, that's activation of oncogenes, and there's loss of tumor suppressor genes. This is caused by radiation, chemicals, and physical irritants. Transport mechanisms. Now, transport can either be number one, passive transport, number two, active transport. So let's start with passive transport. So what's passive transport? Passive transport involves three conditions. You can see no energy required to move substances across membrane. So example is water, lipids, and other lipid-soluble substances. So to make the work easier, in passive transport, there are three conditions that must be fulfilled. Number one, there's no energy involved or required. Number two, substance is being moved from high concentration to low concentration. Number three, carrier proteins are not involved except in facilitated diffusion. Please emphasis in facilitated diffusion. So these three criteria are what it means to be passive. So the types of passive transport we have is number one, diffusion, simple diffusion. We have osmosis, we have facilitated diffusion, and we have filtration. So quickly, what is simple diffusion? Simple diffusion is simply the movement of substances from a region of high concentration to a region of low concentration until a state of equilibrium is attained. So an example of diffusion is seen in the body, in the lungs, where there's exchange of gases via diffusion. Right? So that's it. Then osmosis is the movement of water molecules through a semi-permeable membrane, usually from a region where water is high to a region where water is low. That means it's from low concentration to high concentration, because water is more in a region with low concentration and less in the region with high concentration. So water will move from where it is high to where it is low. I hope you get that, or you need to go over it again. Alright, so an example of osmosis in the body is seen everywhere water is moving. Everywhere water is moving. Then we have facilitated diffusion. In facilitated diffusion is a type of passive transport that is facilitated. It is helped by using the carrier proteins. So example of facilitated diffusion is seen in transport of glucose and amino acids. Then filtration, some classification will put it under bulk transport, but filtration is movement of substances under the influence of hydrostatic pressure. Under the influence of hydrostatic pressure, from a region of high hydrostatic pressure to a region of low hydrostatic pressure. So filtration is seen in the kidney, ultrafiltration is seen in the capillaries. That's where you have filtration. So this is passive transport. Moving on, we'll talk about active transport. Again, active transport is the vice versa for passive. So number one, energy is involved. Number two, transport of substances or materials against concentration gradient. So this time around, you're moving from low to high. So it's like the poor giving the rich. Are we good now? All right. And the third condition is that carrier proteins are involved. So under active transport, you have primary and secondary. In primary active transport, it just means that the energy that is used is gotten from ATP. In secondary active transport, the energy used is gotten from other forms. So under this, you have special transport mechanisms like endocytosis for large molecules. So you have for large molecules, which you call bulk transport, we use endocytosis and exocytosis. Endo, substances are being taken in. So when a large substance is being taken in, first there's invagination, then there's pouch formation and vesicle formation. Now, endocytosis is divided into two. If what is being taken in is in liquid form, it is called pinocytosis. If what is being taken in is in the solid form, it's called phagocytosis. Phagocytosis is called cell eating. Pinocytosis is called cell drinking. Then you have exocytosis, when a large molecule is being taken out. That's exocytosis. Then there's transcytosis. In transcytosis, a molecule is being taken in and out. So transcytosis is a combination of endo and exocytosis. Very, very important. Now, this is transport mechanism. This is transport mechanism. In summary, what have we done? We have said that the cell is the basic unit of life. Cell is made up of organelles. The cell membrane is a phospholipid bilayer following the fluid mosaic model. Organelles perform specialized functions. All organelles are made up or covered by a limiting membrane except for centrioles, cytoskeleton and ribosome. Nucleus and mitochondria are special organelles because they have 1. Double membrane, 2. DNA. DNA in nucleus is called chromosomal DNA. DNA in mitochondria is called non-chromosomal DNA. Transport is divided into two. Primary active transport, energy is not involved. Passive active transport, energy is not involved. Passive transport, energy is not involved. Active transport, energy is involved. Endocytosis, large molecule is being taken in. If it's in liquid form, pinocytosis. If it's in solid form, phagocytosis. Exocytosis, substance being taken out. Transcytosis, in and out. Cell death is apoptosis and necrosis. So cells go through pressure, stress and transformation, just like a diamond. So as you study and grow, remember, refinement takes process. And once you become a diamond, you don't lose your shine. Now that's how this concept should be understood, clear, precise, and exam ready. If this helped you, go ahead, like the video, drop a comment, and tell me what topic you want next. This is Dr. ABM, where you don't just study medicine, you master it.